Right. So moving right along, it's my pleasure to introduce Melissa Haendel, who's currently director for, I want to get this right, director of translational data science at Oregon State University, and also an associate professor in medical informatics and clinical epidemiology at Oregon Health and Science University. And you're still cross-appointed at both places, right?
Yeah, I'm making it as complicated as possible for you to introduce me.
Great. If anything I say is wrong, you're welcome to correct me after. So Melissa actually started her career as an experimental scientist rather than an informaticist, and so how she got from an experimental scientist to what she calls herself now a data translator is actually an interesting story. And briefly speaking, she had some misadventures in data, and including being in a lab that fabricated data, and some of her works get followed by Excel spreadsheets and so on. So really. It was an interesting story that she just related to the students an hour ago. But as a data translator, her goal is trying to integrate data from different sources, ranging from health systems, from patients themselves, but also basic scientists, and how to use technology to bring the data together and generate knowledge from the vast amount of data that we in research has been collecting. So without further ado, Melissa, and her topic will be power to the patients and ontological approach to patient-clinician collaboration on rare disease diagnosis. Thanks.
Thanks so much, Will. It's really exciting to be here, and I really enjoyed our session with the students just before this. You guys have an amazing program here. You're very lucky. Okay, so I didn't know how much background people have, so there's quite a bit in here, and so maybe it's good for the newer students, but many of you are probably already familiar with some of these technologies. Let's see, how do I make it go? There we go. Okay. So, we just heard some nice examples about, really leveraging whole genomes for discovery of new candidate loci for molecular basis of disease. But when we think about more complex diseases or more common diseases, there's actually a whole lot of data that we would really like to be able to better utilize, both on the individual patient side as well as on the population and cohort side. So, we're really kind of just scratching the surface in most diagnostic settings, where we're really kind of just, if we suspect it's a genetic disease, we'll do the whole exome or whole genome sequencing. But many of these other types of data are rarely used, even still, in a sort of production level clinical environment. And similarly, we have population level information. The NOMAD collection has really revolutionized our ability to have reference population frequency, but we don't really have very good reference information for some of these other data types. So how we actually define disease, and I'm going to talk a little bit about that today, but correlations with disease and understanding how phenotypes correlate with all of the other data modalities. But then, further information about, disease risk factors, and really thinking about how our diet, I know some of you work on food ontologies up here, but how our diet and our exposure over time longitudinally interacts with our genomes and affects the outcomes of our diseases. And so, fundamentally, one of the goals that we have is to try to really try to figure out how do we actually better utilize some of this data in order to have better discriminatory power for disease diagnostics, for understanding how to create better prognoses and select the better treatments. Let's see, I'm going to get... So my 4-year-old, who's no longer 4, once said to me that Mama, dogs are a lot like people. And I said, yes, why, son? In fact, they are a lot like people. I thought that was very astute for a 4-year-old. but the question is, how do we actually know that? How do we actually compare the phenotypic information that we visualize, that we observe from one species to another? Do not follow this. So the answer, we also know that if we have more species, we actually have better coverage. And the reality is that we don't actually know the phenotypic consequences of a very large portion of the human coding genome. And so we'd really like to be able to leverage information from those other species. So if we just look at, these were the 19,000, this is going to drive me absolutely nuts. I wonder, maybe? Okay. Sorry, I'm a Mac person. So if we look at the number of human protein coding genes that were in HGNC in April of 2018, there are about 19,000. Roughly only 4,000 of those have phenotypic or disease associations associated with them. So while we might know something about them in terms of what transcription factor binding sites they might have upstream, or those sorts of things, in terms of understanding what do variations in those genes actually cause, roughly only 23%. So, but if we take the orthologs of all of those 19,000 genes and we look at the top 5 most widely used model organisms, and we look to see what we know about the phenotypic consequences of mutation in those genes, we have roughly 15,000 of those genes we actually know something about. And if we look at the union, we actually have 83% coverage of the human coding genome. So we've really gone from 23% to 83% coverage of knowing something about an ortholog in another species for a human coding gene. So that's actually a lot of phenotypic information that we might be able to leverage for the purposes of diagnostics for rare disease. But I'll go back here. I come back to this problem. How do we actually use those phenotype information to compare those phenotypes across species? So I'm going to talk a little bit about that. So I call this the chasm of semantic despair. That's me at the bottom of the chasm. And what the chasm of semantic despair really is the fact that the vocabularies, information, the way that we describe phenotypic features of these model organisms or even our pet species is fundamentally different than the clinical data that's used to describe patient attributes, labs, images, et cetera. And so, we have this fundamental language problem of trying to relate physical feature information in one community or one species to another. So how do we actually help machines understand what these phenotypes mean and how do they relate to each other? So here we might have a clinical term called palmoplantar hyperkeratosis. And the computer doesn't know what this is. It's just a string, right? But what we really want to know is how do we actually relate these to other kinds of phenotypes in other species. So a patient might refer to that term as having thick hand skin, whereas a similar mouse phenotype might be referred to as an ulcerated paw. So it's not a string matching problem, it's a conceptual alignment problem. If we see a phenotype in a mouse we want to understand how might this be representative as a model of human disease, we're just not using the same words to describe those features. So the additional challenge is that each one of these different species or communities, even in the clinical communities in different disciplinary areas, have different terminologies for describing phenotypic features. And there's actually hundreds and hundreds of these different terminologies that exist that are largely uninteroperable. So a number of years ago, we actually took an approach where we aimed to build logical definitions for these vocabulary terms that describe phenotypic features. So here, a human phenotype, palmoplantar hyperkeratosis, would be described using a logical axiom, using terms from other ontologies that were species neutral. So here we would create a logical expression that this term, this string that the computer knows as a string, actually has an identifier, is logically defined as being an increased keratinization that's located in the stratum corneum layer of the skin that's part of the autopod. And these terms over here on the right, the sort of adjectives in Pato, the gene ontology, which many of you might be familiar with, and Uberon and anatomy ontology are all cross-species, all species neutral. Then when we have a similar logical expression or a related one in another organism, we can actually know that these two things are equivalent or otherwise related based on the logic. So we were able to show this back in 2009 that a, quote, phenotype BLAST, it's not really at all related to the BLAST algorithm, was able to actually retrieve the correct mutation in different species when we represent those phenotypes using that atomic approach and that logical approach. And so this was quite exciting because it was the first time we were able to sort of compare phenotypes across species and reveal the underlying molecular basis of the phenotype from another species. So I'm telling you all this because I want to show you a little bit how the algorithms work, and then as we go towards the end of the talk, how we're going to get patients to actually participate in this. So if we think about, this is a very simplistic view, but if you think about a patient or a disease being described in part as sort of a collection of phenotypes, and you kind of think about it as a bag of phenotypes. Now, of course, these different phenotypes have different onsets, different expressivity, and they change over time. But just for the purposes of this talk, we can think about this patient at the top having a collection of phenotypes described like a flat back of head or hypotonia, and we're trying to understand how does, it's a matching problem. Your patient comes into your clinic. How well do they match what we know about the collection of phenotypes that are associated with a disease? So here we might have a disease that's defined as having an abnormal skull morphology and decreased muscle mass. And the clinician by Gestalt fundamentally knows that having a flat back of the head is an abnormal skull morphology, and hypotonia is related to decreased muscle mass. But it's not, again, it's not a string matching problem. So there's kind of fuzzy relationships. And these of course are quite complex if you think about all the different variables. So as part of this effort to build the Human Phenotype Ontology, we have an ontology that has these different logical expressions, and they're annotated to over 7,600 diseases, known Mendelian diseases now, some common and complex diseases as well. So so if you look at the hierarchy of the Human Phenotype Ontology, the less specific terms, abnormality of the orbital region, would have a subtype, abnormality of the globe location, which would have a subtype, hypertelorism, and the number of diseases that are annotated with that term goes down as you get more specific in the graph of the Human Phenotype Ontology. And so, let's see, so we have over 13,000 terms in this vocabulary and over 155,000 phenotype annotations to known diseases. So the goal is to really then describe the patient using nodes in this graph, and this is a little bit silly of an animation, but it's still yet to date the best way that I can describe how the algorithm is comparing things. Maybe some of you have used similar types of algorithms for graph operations. But so here the patient is described as having nodes in the graph where the different colors represent different anatomical systems. And we're trying to understand which disease or which model system might be most similar to that patient that's been described. And the situation with a rare disease patient is that, we don't have that gestalt. It's not about knowing that, the flat back of the head is similar to abnormal skull morphology, because the clinicians may never have seen any patient like the one that they have sitting in front of them. So the goal of these algorithms is to help reveal what some of those similarities might be. So, this fuzzy phenotype matching looks for the graph, and that didn't translate very well, but looks for the graph that's in common. So what are the kind of least common subsumers, and sort of taking breadth and depth and annotation frequency into account, what are the most sort of similar graphs that we have? And there's a whole family of algorithms that help do this, and we've implemented some of them in the LSIM package, which I'll give you a link to later. So, so this may be of interest to you for those of you who are doing kind of graph-type operations, but it allows you to use different algorithms for doing graph-based comparisons. So the other problem is that what we hang these phenotypes off of, these diseases that we're trying to match, there's actually many different resources out there that create these disease definitions and annotate phenotypic information to a given disease or genotypic information. And when we look across the landscape, we see that this is a combination of the OMIM, MESH, Orphanet, and the disease ontology. And so when you combine them all, you can see that there's sort of this crazy logical chaos of cross-referencing and subclassing across all these different vocabularies. And the problem is that when you actually combine all the data that you need to make the diagnoses, to make the software for the diagnoses work, you end up with these terrible inconsistencies. So if you follow everybody's assertions, everybody's quote mappings, and I truly have nightmares about mappings on a regular basis, that you end up with these inconsistencies. So here, atypical hemolytic uremic syndrome is basically inferred to be equivalent to typical hemolytic uremic syndrome, which is a really terrible thing. If you've got a patient sitting in front of you, you're not exactly going to be able to give them the proper diagnosis. So, so we have some work to go, that had an algorithm that goes, that's called KBOOM, which I forgot to put the link in, that takes all the different XREFs and a bunch of priors that does string matching and synonym matching and kind of puts it all in a big hopper and says, okay, what are the most likely parsimonious equivalence cliques? And then for the ones that have low probability, we go through and we curate them. And this has been a collaboration with ClinGen, where we also have, similarly, in combination with that algorithmic approach, a robust definition for when we create a new disease and how we actually understand what a disease definition should be. So does it have a distinct molecular mechanism? Is there an assertion of difference from other known diseases? Are there different clinical management strategies? And are the phenotype profiles fundamentally distinct, which we can measure with graph metrics? And so this has all been pushed now into a brand new unified resource. We just call them Monarch Disease Ontology for lack of a better label. So there's many other kinds of data that have been integrated, and I just wanted to briefly mention this since a bioinformatics audience might be interested in some of our APIs. And in this case, we've really taken a lot of different species and different resources and combined all these different data into one large graph store. And what's interesting is that actually when you create the genotype to phenotype associations, you can get almost over 90% of our associations that we have in our corpus require integrating more than two or more data sources. So we have this real capacity to infer candidates based upon these harmonized data. And it's also used to drive a variety of different types of analysis tools and interfaces. And so just for later, there's an API. It does a lot of variety of ontology-based types of operations. So you can look for all these different types of assertions across different data modalities, and there's some literature features in there as well. Oops. So it also allows us to drive different types of data visualizations, and I also put this in here, and we didn't quite get to this in our earlier conversation, but one of the real challenges that we have in our community of sort of semantic engineering is we have really terrible ontology browsers and ontology visualization tools. So this is actually a call to action, and I'm hoping that one of you will be a genius in sort of semantic visualization technologies in your future. And so these are just two of the approaches that we've come up with. We're not actually very good at data visualization, but one of them on the left there aims to sort of compare phenotypic similarity across sets of phenotypes from the patient or the organism of interest on the y-axis against very many different species and diseases on the x-axis. And the one on the right aims to try to provide a sense to the clinician for how similar are two different disease profiles and give some metrics around that. I recently at the ICPO conference gave a rant about ontology-based visualization challenges. So if any of you are really interested in data visualization and need a new project, maybe you can go there and find a problem. You can see the links later. So just a little bit about our Exomiser analysis tool. We heard a little bit earlier about, how exome analysis has become quite commonplace. But again, it doesn't necessarily leverage phenotypic information as much as it might. And so this tool that we've developed called Exomiser leverages the phenotype comparison algorithms that I just mentioned, but it also leverages protein-protein interactions in order to support sort of guilt by association across different species. So we're also leveraging the protein-protein interactions coming from different species. And so when you combine these two different approaches, you can end up with a much smaller set of candidate variants when you do variant prioritization for your rare disease patients. So this is an actual example where we had two different patients in our clinic. Over there on the left, a 3-year-old girl, and on the right, a 14-year-old boy. And the clinician didn't recognize these two patients as being at all related to the same disease until they actually used our tooling to show that their phenotypes were actually similar to our gold standard curated disease definition, which is there in the middle, which is Wiedemann-Steiner syndrome. And so in this case, you can see that the patients on the right had some phenotypes that are actually opposite, and we've been doing some work lately to look at how do opposite phenotypes relate to the genotypes that we have in hand and our disease definitions. But here, macrocephaly in the 14-year-old boy and microcephaly in the 14-year-old girl. And it turns out that both of these patients, within just 2 weeks of each other in the same clinic, but two different families, had two different variants in the same gene, and also really kind of gets after how do we actually define diseases. So right now, this is still Wiedemann-Steiner syndrome, even though there are two different variants in the same gene. They clearly have very related phenotypes, but in some cases opposite. So this is a lumping and splitting problem. So how many of you are bird watchers? Any bird watchers in the audience? So are you lumpers or splitters? I'm a lumper when it comes to bird watching and a splitter when it comes to diseases, as it turns out. So this is another example case that we helped solve by using Exomiser, where based on phenotype similarity to a mouse where there was no known data in the human databases at the time, we were able to prioritize a variant in STIM1, and this family was diagnosed with a mutation in STIM1. And then one of our consortial members is part of Genomics England, so Exomiser is being used on all the Genomics England 100,000 Genomes project. And of the first 1,900 of those, 82% of them have come to the top 5 using the tool, using this phenotyping and genotyping together approach. So that's really exciting for us because it's been many years since we've been building these ontologies to be able to do that. And then similarly, we've taken the 263 hardest cases in the 100,000 Genomes project and have seen also really good performance from Exomiser by using phenotype data. And this is an example of one of those families. So in this case, Jessica was a 4-year-old who had a rare condition which causes epilepsy and affects her movement and developmental delay. All the standard tests were negative. Exomiser ranked this variant first, and you can see kind of over the left how it sort of works out in the clinician's minds. It starts out with 6 million variants. Over 600,000 of them are rare variants, and over 2,000 of them are predicted to cause a change in a protein. And 67 of them were different to her parents. So narrowing it down to just what to look at is quite a challenge. And so in this case, one of them rose to the top. And actually the really cool thing about this family is that it's a de novo deletion in SLC2A1, and it's being treated with a ketogenic low-carb diet. So this is one of those wonderful successes where we would have lost this child if it hadn't been for a diet change, right? So we love to tell that story. So how can, now to the actual topic of the talk with all that background, how can patients and families be empowered to help with all of this process? So it's really, now's the time more than ever for this to happen. So it's quite challenging to get all the phenotype information structured and provided to these algorithms. So what a better way to have the patients be engaged in the process of their diagnostic odyssey than to collaborate with their clinicians on the phenotyping. Often, physicians have very little time with their patients. We really need this continuity between physicians and patients in more than just these short increments of their visits. And oftentimes, patients have phenotypes or are otherwise knowledgeable about their symptoms in ways that clinicians are just simply not going to ever observe, like a baby who's inconsolable or who doesn't cry tears, or snoring, or things that don't happen in clinical observations. And so these features are things that are much more atomic than what are normally measured in the clinic in some cases. So really thinking about the patient as a biological subject rather than a billing subject, as so much of the EHR is designed to do. So, and with the increase in, patient wearables and all the different types of measurements that patients can be, generating now, it's really the time more than ever to think about data streams that come directly from the patient. Plus, for the rare disease patient, it's an opportunity to educate them about what we know about what we're finding about their symptoms, even if it's from another disease or another gene. It's something for them to really think about and hang their hat on, and which is really important for them. So so we recently we've translated the Human Phenotype Ontology into a number of different languages, but we recently translated it into laypersonese. And so this is just an example of some of those terms. So we might have, a term micrognathia, which we would have a layperson synonym for small jaw, or arachnodactyly, which we might translate to spider fingers. And so really trying to make the terminology accessible to the lay patient. And so we looked here at after we did this for the whole ontology, so it's a long, laborious web searching exercise to add all the synonyms. We started to look at, well, how does our coverage of known diseases look, given if we just look at the lay subset of that ontology? So roughly 4,500 HPO terms have lay synonyms, and there's around 7,600 synonyms total. So that's about 35% of the ontology, and it's about 60% of our 7,600 or so diseases actually have annotations to a lay term. So that's kind of the background for this study. So one of the questions is, how many terms can you take away and still recognize the disease? So patients are not likely to have all the clinical terminology, and they're also likely to exhibit, missing like the phenotypes that are subtle or they don't understand, or have noise because they have other kinds of features or that they might have. And so the question is, how can we help patients create the most robust profiles that are diagnostically useful? So, one question is, how much phenotyping is enough? And so we have some algorithms that help support, an understanding of what's the uniqueness, what's the breadth and depth. And here in this example with our favorite Star Trek species, I also studied craniofacial development, so it's kind of a double whammy here. We have these phenotypic features like hair being present on the head is not a very unique phenotype. Being male or female is also not very unique. But if we're really looking for, robust phenotypic profiles, that one in the middle, increased skin pigmentation, enlarged lip and enlarged ears, aren't necessarily each individual phenotypic feature is in itself unique, but the combination is robust in the face of all the other profiles. And so the algorithm measures that sort of capability, because at the end of the day, these are new phenotype profiles being put in by patients or clinicians, and so they're not. We're not saying that what you've given us is actually diagnostic, but just trying to get a sense for, like, when is enough phenotyping? We actually applied this in our clinical tools because the clinicians really didn't like manually having to enter all these terms. And when we started giving them gold stars for getting high annotation sufficiency scores, the amount of terms they gave us actually went up quite a lot, which just shows you the power of a gold star. So how diagnostically useful are the HPO terms generated by patients? So there's a survey that's actually the standard of care in the U.S. for undiagnosed diseases in the Undiagnosed Disease Network, which is the Genome Connect survey. And that survey mapping has a few hundred terms mapped to it. Then we have our lay HPO, which has about a third of the HPO, around 3,500 terms, and then the clinical grade HPO, which has around 13,000 terms. So we would imagine, of course, that the diagnostic capability would depend upon how many terms we actually have access to. So we actually just looked really quickly at some comparisons. So here in the middle, this is for nephroblastoma, our gold standard set of HPO annotations for that disease. And you can see the lay synonyms over there on the left. So, some of them, of course, the same. Abdominal pain is the same for layperson as it is for a clinician. But, we might have hypertension, which is high blood pressure. And so we have quite good coverage. There's only a few that are not quite exact. But the Genome Connect simulated profile, you can see you have much higher level terms. So, nephroblastoma maps to, in a fuzzy way to renal neoplasm, or aniridia maps to abnormality of the iris. So they're much higher level terms that have been mapped to this survey instrument, and that's because the questions are things like, do you have any abnormality, do you have any abnormalities in your iris? It's not actually specific to the more descriptive clinical phenotype. So for those different diseases, we compared the sort of diagnostic power of those diseases. So, okay, I'm going to have to use this. Let's see. And so basically for every known disease, we created a simulated profile for the Genome Connect survey terms or the lay HPO terms. So, for every disease, there would be kind of a subset disease that we would then use as the query against all the other 7,600 diseases to see which ones would result in the best ranked disease being returned. So essentially, back to your face picture, can you still retrieve the correct disease when you're missing a portion of the profile? And then we also added noise, so adding extra random terms and subtracting extra random terms, and used a couple different algorithms to do the comparison. And so here, for our gold standard annotations, roughly 7,600 diseases. For the LaySlim simulated profiles, roughly half of them or so recalled the correct disease within the top 5 hits. And then, of course, you can see that the Genome Connect profile doesn't perform quite as well. We also found that the Bayesian comparison algorithm, BOCA, performed better than our kind of more standard Jacquard-based similarity algorithm in this case. So that was actually pretty amazing in the sense that we take away 2/3 of the ontology, and we still have the ability to correctly rank the right disease in the top 5. So that suggested to us that, in fact, patients should be able to provide robust phenotypic information. Even though it might not be quite at the level of a clinician, it's still informative information that the clinician can use in their diagnostic tools and in their assessments. We also just looked at this comparison of the LaySlim profiles against Genome Connect to see, what was the difference between those two, and 95% of the time, the diseases for the lay HPO outperform the Genome Connect, but not 100% of the time, and we were quite surprised by this result. And so why are there some gaps? So one is that some of the existing HPO terms that should be a layperson, like schizophrenia, were not actually designated that way. So we had things that were mapped in the Genome Connect set that probably should have been mapped as layperson. There are some terms that really aren't layperson or borderline, such as abnormal EEG. And then there are some things in the Genome Connect survey descriptions that just don't really distill into lay-friendly labels, and so we weren't really able to translate those ones. So that's kind of interesting. We also looked at disease enrichment as well as disease non-enrichment, anti-enrichment. And, you know, it makes a lot of sense. The diseases that are enriched are most diagnosable with the lay HPO are things that are physically observable. So you see things like bone diseases and skeletal, congenital limb malformations and things like this. Things that were anti-enriched, I don't have a slide for this, are things that are a lot of things that needed molecular characterization, so things that you need clinical laboratories in order to measure. And so that makes a lot of sense and really gives us a sense for how can we build the best tools for patients, knowing that they're obviously not going to have you know, ready access to that information, although they may have it if they have their own clinical records. So we have a new tool that we've been developing called Phenotyper. For lack of a better name. If you have a better name, I'd love to hear it at the moment. And it's being launched in patients next month. And so it's just, it's in its very early days, and the goal is to actually understand from the patients, we've limited it so that they collect information about each anatomical system kind of in an autocomplete fashion on the ontology. And it's a very simple interface. I'm not even going to show it to you because it's that simple. But the idea is really just to test how patients interact with the ontology so that we can understand how do we build a clinically validated tool for use by patients to provide data with clinical CLIA certification. So the idea is that they were going to compare the standard survey instrument against this ontology-based approach to see how well the patients are able to provide their phenotypic features. And these are all diagnosed rare disease families, so we'll be able to compare them against the gold standard. I wanted to mention just a couple things really briefly. This is somewhat unrelated, but some work that we're doing in the Global Alliance for Genomics and Health, which is a standards organization for genomics, is to actually build a new standard for exchanging phenotypic information. So we have standards for genes. We have no shortage of them, in fact. We don't really have very good standards for how we shuttle environmental or phenotypic information around. So this is a new effort that we've been working on in collaboration with people who work on FHIR technology for EHR data exchange, and trying to understand how do we get the tidbits out of the EHR and combine them with other tidbits that the patient might give us in order to have the most robust phenotypic packet that we can use in these genomic diagnostic tools. And this data structure can be used in other contexts as well. It can be used for the clinic to send information to a clinical lab. It can be used by journals. It can be used by patient registries. And so if any of you are interested in this kind of a problem, I really invite you to participate in the Global Alliance. It's actually a great global community. So what's next? What comes next with our project? We're working now on sort of how do we improve the tools so that the patient can have real-time feedback based on the annotation sufficiency. So, you know, we need to figure out better how do we do that, the gold stars for the patient-centered lay at HPO like we have done for the clinical environment. And then, you know, the study design that we have for this study that we're undertaking, you know, really aims to have those patients provide full profiles so that we can actually assess how well they do. But in the reality of the case, and that's needed for clinical validation for CLIA certification, but for you know, for the clinician who really just needs to know the most important things from the patient, we have other algorithms that can ask questions like, what's the most next valuable question to ask? And we can provide smart logic using the ontology to suggest more narrower parts of the ontology. So if a patient annotates themselves as having X, Y, or Z, then the tool should suggest something in the area of W, right? Because W is commonly co-occurring and is diagnostically useful. So these are some of the things that we're working on now in terms of a future application. So just to conclude, so ontologies can be used to harmonize phenotypes across patients, diseases, and different species, and can aid disease diagnosis. We are dedicated to the idea that patients are a key source of phenotypic information, and that the clinicians and the patients should both need to participate in phenotyping to maximize their diagnostic capabilities. For over 50% of our 7.6 thousand diseases, the correct disease was identified in the top 5 using the lay HPO, which suggests that a layperson translation of the HPO is diagnostically useful. And as I suggested, we're going to be validating for clinical use in patients and hopefully hiring a gamer to make this tool really fun for patients to participate, so they really want to give us lots of phenotype data, and also add in the phenopackets export. So I really want to thank Kent Shevchuk, who's been leading all of the analysis work, Nicole Vasilevsky, who did all of the layperson tedious work of translating the HPO into laypersonese, and to Kat Brownstein, who's helping collaborate with me on the development of the tool, and Ingrid Holm, who's running our patient study. And I should also mention Chris Mungle, who developed all the algorithms for doing the graph matching. And then finally, but not least, hopefully it's still there. Oh, did we download this? Or is it running live?
I think it's live.
I added a slide at the end.
Oh, yeah, you're going to have to reload it. Maybe hit escape.
Oh, let's see. That's what you get for editing your presentation while the first speaker's going, right?
Reload the page, that should be fine. That's fine. It's not coming for another 15 minutes.
Let's see. There we go. The most important slide, calling all parasites and symbionts. So there's the Research Parasite Award being given for people who reuse other people's data. So I know that all of you reuse other people's data because you're bioinformaticians. So you can actually, if you do a really good job of it, you can get an award, and the award is a lamprey. So it's really exciting, and we really don't have enough applicants, and there's some junior awards for students. So I think it would be a wonderful thing for any of you to have on your CV. So please nominate yourself or one of your friends. So here's the criteria for the award. You must not have been involved in the design of the experiments that generated the data. You have to have published independently of the original investigators, and the original investigators are not actually authors of the secondary analyses, but are appropriately credited in the manuscripts, although I think that could be negotiated depending on your justification. The awardee may have extended, replicated, or disproved what the original investigators had posited, and the awardee has provided source code and intermediate or final results in a manner that enhances reproducibility. And as a member of the award team, I do not have enough submissions right now, and so I am going to be in deep trouble if I go back after my conference here with you all and don't get at least one submission. So please, please, please. And then they also will, I believe, fly you to Hawaii for the biocomputing conference in Hawaii in January. So there's that as well. So yeah, please apply for the Research Parasite Award. So thanks very much. Questions? Okay, I can try. Yeah?
So I'm kind of interested, how did you, you said there was varying descriptions for when doctors were phenotyping their patients. And how did you relate those again? Were you using NLP, or was it manual, or?
So the question is different phenotypic descriptions from different clinicians, and how are those phenotypic descriptions related? And it was at manual or NLP. So we actually have both approaches, and we found that if you really want to do a robust job of capturing the phenotype terms, that a purely manual approach is quite laborious, which is part of the reason for getting the patients involved in the first place, is that can actually help, reduce the burden on the clinician for doing that. But we also have some ontology, kind of more specialized ontology NLP tools that will mine sort of clinical notes or phenotypic descriptions. And I was just talking with Wyatt this morning about some tools that you all are working on here to try to do audio, audio transcripts that could be used, and so I think there's some opportunities to use that as well. But basically, the tools capture the HPO terms, whether it be via text mining or manual, resulting in a set of identified terms that then the graph matching algorithms are used. And so in that example that I gave, they actually, it was the same clinician who didn't even realize it until they looked back at their patient that they had had 2 weeks earlier. So that was like phenomenal, right? Because that's like internal control, even not even just a different clinician. So, yeah. Yeah.
One thing people seem to worry about is like in the area of direct-to-consumer genetic testing, people getting these tests, the doctor saying, oh, I have cancer. So what would happen there? How you looked at your tools, like if somebody kind of just went and used the tool who wasn't kind of maybe sick, the doctor would say, but they say, well, I'm irritable, I'm gaining weight, and maybe they're just in grad school. Like, kind of like overdiagnose people?
That is a really good question. So the question or concern is about, with all the direct-to-consumer tests that are out there, are patients going to be sort of overdiagnosing themselves? I think that that's true right now, even without good tools, is that we're all really good at searching the internet and figuring out what problems we really don't have, right? So I think, so that is a challenge, and it is an ethical concern. I think, we're starting, pretty slow here in the sense that the goal here, these are patients that come in with undiagnosed diseases, and so they're absolutely going to put stuff in there that's they have a headache because they drank too much, or they are lethargic because they don't exercise enough or whatever, right? So those things are all going to be the noise that we have, but our hope is that the signal will come out in that context, and that we'll also learn better how to understand what that signal to noise looks like so that we can grow these tools to more complex and more common diseases. And we definitely are collaborating with some other groups that really want to do this at scale for anybody doing genomics. And I do have concerns about exactly your point, but I think that validating first in this kind of a setting will help educate us on better approaches to help reduce that. And I think it's also has to be combined with educational information, so that right now the tool is really simple because it's just testing one thing. But if this were to go live for anybody getting direct-to-consumer genomic testing, we would want to have an enormous amount of educational materials available in that kind of a context to try to reduce the sort of worry factor that patients would have from using such tools. So, yeah.
Great talk.
Thanks.
It's amazing to see how the work has progressed since the last time we saw your talk.
Oh, thanks.
And so the one sort of standard and terminology that you didn't really address in your talk and that I'm struggling with is the clinical terminologies that are used in the electronic medical records like ICD-10 and SNOMED. And so how do we map from there to this fantastic ontology that really allow us to work and mine the data?
That's a good question, too. So the question is, struggling with how do we map the currently used clinical terminologies to the HPO and use these types of technologies in a more interchangeable manner. And that is a problem that we are working on also. So I can give you sort of different answers for different terminologies. So for SNOMED, we actually have a collaboration with SNOMED, and we're going to be creating a integration strategy so the HPO will actually be released within SNOMED in some fashion. And so that probably would happen within the next year. We're actually, the technical teams are meeting in like 2 weeks, and there's some licensing issues, so that kind of gets back to some of this conversation we're having with students, because SNOMED isn't licensed in an open manner, whereas HPO is, and so we're going to have to figure that out. The problem also there is that SNOMED is designed and structured very differently and doesn't actually contain the level of phenotypic features, which is of course why they don't have the content, nor does it really contain the kinds of disease definitions that we have that we need for genomic medicine. So they're really going to have to not just adopt the HPO, but actually also our disease ontology or some disease definitions that are more aligned with genomic medicine as well. So there's a lot of both scientific and technical challenges that have to be addressed, but I think there will be something available in that one. Let's see, what else? For the NCIT, so HPO really doesn't do cancer. We've been working a lot with the NCI to integrate NCIT with HPO and Armando disease ontology, because some of it's more disease level information, disease definitions. And that's going really well, and that we've just convinced NCI to make their ontology CC BY. So it's now openly available for redistribution, however you like. And so that's really changed the landscape. So a lot of communities hadn't really been using NCIT because it didn't have an open license or a readily available license. Plus, it was also not very interoperable, so we've been rewiring all the logic with the same underlying logic that I showed for the model organisms. So, you know, the anatomy is all consistent between NCI's cancer definitions and the HPO's, you know, anatomical locations, for example. So fairly soon there will be kind of a suite between NCIT, HPO, and Mondo that will have coverage of both cancer domains and sort of, you know, Mendelian dysmorphologies and the kinds of things that HPO better covers. As for ICD is just bonkers. I don't know about that one. We don't really map to that. It is mapped to HPO in the UMLS, but those are not, you know, mappings that I would necessarily trust to use in a diagnostic setting. The ICD-11 effort is underway, and that effort is much more semantically rich. And so I think when ICD-11 comes out, we'll be collaborating with them, and it will be a more rich way of representing things. But the way ICD is designed, it's really not designed for the kinds of semantic algorithms that we would try to use, because it's all single inheritance. So yeah. So I mean, I think. Yeah, I mean, we have two. You can certainly use the UMLS mappings or the mappings that we have to crosswalk, but it just would require a lot of manual review. That's the one I just don't quite trust. But yeah, so that was a long answer, but I know you needed it, so that's okay. Yeah.
So two questions. The first one comes from a student group in the Okanagan who's watching the webcast.
Okay.
And that question is, are these software just for an ontology in the English language, or is there any plan for projects to kind of extend into different languages? Because rare diseases are not a uniquely English problem. In fact, some of the cases we have come from, you know, overseas. And so is there any kind of plan to extend, especially your layperson kind of phenotyping into languages that actually relate to several of the patients we actually see?
Okay, so the question is, you know, do some of the tools that we're developing function in other languages, and, you know, how are we going to sort of make patient-centered phenotyping a reality for patients that whose native languages are not other than English? So there's a couple things to talk about with regard to that question. So first of all, we do have the clinical HPO has been translated into many different languages now. It's a volunteer effort globally. We're trying to get as much help as we can, but it's, you know, it's slow going for some of the languages. But some of the translations are quite robust. We have an excellent collaboration with the Japanese undiagnosed group, and they've translated the whole thing, and they're adding layperson terminology now. So that's been going really well. The Spanish translation is also really robust. So there are some that are kind of further ahead. Since don't natively speak any of those languages, it's only by collaboration and partnership that we can achieve that. And the technologies that we have for doing those translations are not ideal. One of the challenges there, and so this is an informatics problem, so if anybody wants to work on this tool, it's another ontology tool problem. Right now we just have this kind of line-by-line sort of tool that's an open source tool that just sort of lets people translate things from one to the other and then keeps track of the provenance. And the provenance is nice, and it's a perfectly reasonable tool. But what we don't do very well is we don't have the ability to sort of push things from one language to the other, or to leverage the graph structure itself to help with the translation. So, really what you would want in a translation is to be able to see the local graph structure in your language in order to make the right synonyms, whether they be lay synonyms or not. And then also when somebody wants to add a term in one language, that we propagate it back to the others, right? So that there's this synchrony across all the different translations. And so we really lack for good tools to be able to do that. The other thing that I sort of thought about talking about, but was maybe not enough time, was also just having patients share their N of 1 cases. So we're also working on kind of a variant file that is sort of phenotype features profiles using the phenopackets with candidate variants that can be shared as sort of N of 1s, and then we make those available alongside our disease definition files so that the algorithmic tools can utilize those candidates without having to wait for something to be published in the literature. So, we talked earlier in the student group about, preprints and getting things out there. This is another example of one of those things where, how much we can expedite a patient in another language being able to find the N of 1 because they're using the same tooling in different parts of the world, and they find a patient that's on the other side of the world that has that same disease, a similar related disease definition. It just really reduces the diagnostic odyssey time. So that's, and those tools, some of those tools are being built in those other languages to help support that. And, but we, again, lack for tools for being able to manage it as well as we would in other languages. Sorry. So it's a really hard problem, and it's something we're really dedicated to. We just need better tools and more collaborators. So, yeah.
And my question is that Exomiser and even Genomiser, to that effect, takes a very single gene view of Mendelian disease. But the reality is that 2% of the genome is coding, and the vast majority of the genome could harbor, genetic variants that could take out regulatory elements. And so things like ClinGen and Decipher have been mapping these regions of the genome where if you delete a completely non-coding region upstream of a gene, it's associated with a set of phenotypes. So do you have any plans to extend the current Exomiser view of, Mendelian disease to more of something that captures the entire genome?
Okay, so the question is about including sort of regulatory variants and sort of more complex variation that's not just a single, gene variation in our exome analysis tools. So I think a couple things. One is that we do have, Genomiser does have regulatory element assessment in it as of, I think, last fall. So there's some work that we've done in order to, we curated a whole bunch of regulatory element variations from the literature and put those in there and did some, not my group, but Peter's group did some difficult kind of analyses to try to show where we had such few data points, how would you actually prioritize a variant in a regulatory region based on just having these very few set of regulatory variants. So those data are available and they're out there. We also are creating new disease models. So this is a longer ways out, but new disease models that take into account a variety of things on both the genotype and the phenotype side. So how do phenotypes change over time, as well as what are the causal relationships between them. And so that's like sort of the complexity that we want to go after on the phenotype side. And on the genotype side, how is that associated with, a more whole genome approach? So the disease definitions are evolving to be more complex on both sides. But creating the corpus for comparison takes a long time to build. So I mean, it's like, I think if we had, better ways of maybe leveraging, what we know about those genome variations, and we actually are working really closely with ClinGen and a variety of these other groups to create these disease definitions. So those are all going in to that same file, really. But, if there could be more automation in how we find these associations and then use those as candidates for creating kind of stub diseases, if you will, I think that approach might expedite things, but we haven't really played around with that yet.